All right, hello everyone. Today we will be covering chapter 9, which is over the muscular system. And as always, we're going to start out with roots, prefixes, and suffixes that you will need to know. So we'll be covering these as we go over the lecture for this chapter. All right, so we have three types of muscle tissue in the muscular system. I think you all know this by now. Skeletal muscle, it's what's attached to bones. It's what makes us move. These muscles are considered voluntary muscles. We have control over those. That's what makes us walk and talk. It's consciously controlled, meaning that we have control over moving those muscles. Cardiac muscle is obviously what makes up the muscle of the heart and the wall of the heart. We don't have any control over how fast or how slow our heart beats, so we call that involuntary muscle. Obviously, it's what is responsible for the pumping action of the heart. And also we have a smooth muscle. Again, we don't have any control over smooth muscle. So again, we call that involuntary muscle. We find this pretty much inside of our body in like internal organs, in our digestive tract, in our blood vessels. So again, not stuff that we have control over. The majority of this chapter will be on skeletal muscle, and we'll briefly talk about smooth muscle and cardiac muscle. So for skeletal muscle, we have over 600 skeletal muscles in our body. Those are attached to bones by ligaments, or I'm sorry, by tendons. Remember, ligaments connect bone to bone, and tendons connect muscle to bone. Again, it's under voluntary control. We have conscious control over walking and talking. And muscle is considered an organ. And it says here, one of the functions is to aid in pumping blood. Now, we said that cardiac muscle also pumps blood, or the wall of the heart actually does the pumping of the blood. But how do you think that skeletal muscle aids in pumping blood? So when we walk or when we contract our leg muscles, we have blood vessels all throughout our body and interwoven in all of our muscles. And when we contract our skeletal muscles, what we're actually doing is we're actually forcing that blood back up towards our heart. So our skeletal muscles actually help in pumping blood back towards our heart. Also the same thing when we breathe. When we breathe out, when we breathe in, our thoracic cavity expands, and when we exhale, the pressure in our thoracic cavity decreases, which also allows for blood to help return to the heart. Skeletal muscle has obviously skeletal muscle tissue, but we also will see that there's nervous tissue, blood vessels, and connective tissue involved. So this next slide, you're going to have to know this slide in and out. From smallest to largest, largest to smallest. And in Canvas, under the module section in chapter 9, I put a document out there that will kind of break down this a little bit more. I have definitions as to kind of what each of these are. So kind of starting out, we have our bone here, and tendons connect muscle to bone. And then we have this layer of fascia. So this muscular fascia is kind of like a branch off of the tendon. And then we have kind of three different layers of tissue that will kind of surround different parts of the muscle. We have epimysium, paramysium, and endomysium. So epimysium, we'll talk about these later on, but epimysium surrounds kind of like the whole muscle, the muscle as a whole. Paramysium will surround individual fascicles, and endomysium will surround individual myofibers. So we have these things. If we, here's our kind of whole muscle tissue as a whole. And if we kind of pull out one of these kind of little circular objects, those are going to be called a fascicle. And so paramysium will surround an individual fascicle. And we'll see that that they have contained within these fascicles the sarcolemma. We talked about like a cytoplasm when we talked about the cells. So a sarcoplasm is just like the cytoplasm of the muscle. The sarcolemma is kind of like the cell membrane of a muscle cell. We also have these things called myofibers, and myofibers have different names. Myofibers. We'll get to these later on in the lecture, but we'll have different names for these. So you can see that if we pull out kind of one of these little circular things, that's going to be a myofiber. And within that, we have the sarcoplasmic reticulum. It's almost like an endoplasmic reticulum. We'll talk about that when we talk about skeletal muscle contraction. And then if we pull out one of these little circular objects, that's called a myofibril. Not to be confused with a myofiber. They're very different. And we'll see that there's going to be a sarcomere. We'll talk about what a sarcomere is, what a transverse tubule is, and then we can pull out these filaments called actin and myosin. And again, we'll talk about what all of these things are. So become very intimate with this slide. You're going to have to know this slide. So use this slide in conjunction with the other document that I put out on Canvas. Learn these definitions, know what this slide is. There's going to be a lot of quiz questions, a lot of test questions about this slide. All right, so here's just kind of like a microscopic view of skeletal muscle. We don't have that epimysium, but we do have that paramysium and that endomysium. Remember, two out of those three layers. We have those fascicles, a muscle fiber, the myofibrils. You can see a nucleus here. All right, so those three, we have connective tissue. We already talked about tendons. Remember, tendons connect muscle to bone. Tendons basically are just kind of like cord-like masses of connective tissue. Provides movement, provides flexibility. We also have fascia, which is basically a thin covering of collagen. Remember, collagen's a protein-based structure that it's very rigid, and it surrounds those muscles, organs, and bones. So if we go back to our slide here, that's our muscular fascia that comes off of the tendon. And we also have this thing called an aponeuroses, or an aponeurosis if it's singular. And that's just a broad sheet of dense connective tissue. Again, collagen, protein fibers. And basically it connects again, muscle to bone, or skin or cartilage, or to another muscle. Again, collagen, strength, stability. Anytime you see collagen, strength and stability, very tough. And we see aponeuroses. Remember that sheet, a broad sheet. If we do some dissection of your gut, we remove that outer layer of skin and adipose tissue, we'll see this big aponeuroses kind of right underneath that adipose layer. It's a big sheet of dense connective tissue, and then your tendons are going to be more cord-like structures. All right, so the connective tissues and the surrounding the muscles. Remember, we have those three: the epimysium, perimysium, and endomysium. You're going to have to know those, and I just put them outer to inner. Epimysium surrounds that whole muscle. It just lies underneath that muscular fascia. Remember, epimysium underneath that muscular fascia. So actually what I will do is we'll do this muscular fascia. There. So when we go back to this slide, lies beneath muscular fascia. Very coarse collagen fibers. So again, strength, stability, remember collagen, protein. Perimysium surrounds those fascicles within a muscle. So perimysium surrounds those fascicles. Again, collagen based. Endomysium is going to surround those muscle fibers. Remember, I said those muscle fibers have a lot of different names. So myofibers, muscle fibers. So now we have two names. And it's actually not collagen based. It's actually composed of loose areolar connective tissue. So that's going to be the only one that endomysium is going to be the only one that does not have pure collagen fibers associated with it. All right, so we talked about that muscular fascia earlier, so we'll do again muscular fascia. Basically, it connects that subcutaneous fascia that lies right underneath the skin. And it also has, it connects that fascia of serous membranes. And what it does, it kind of separates muscles and blood vessels and nerves. And we have this thing called compartment syndrome. If you do a lot of exercising, you might have heard of compartment syndrome. And what compartment syndrome is basically you have fluid accumulation in some compartment within one of your muscles, and there's no place for that fluid to go. And what happens is there's because of that increase in fluid, you have a lot of increase in pressure within that compartment. Because of that pressure, because of that increased pressure, nowhere for that fluid to go, decreased blood supply. Decreased blood supply means you don't have that blood supply to deliver oxygen and nutrients to that area. And it's very painful, and your muscle tissue can start to die without medical treatment. And it's actually a medical emergency if you do have compartment syndrome. And I've seen it once when I worked in the emergency room. And what happens is we have to do what's called a fasciotomy. And basically we have to slice open that muscular fascia layer. And it's like I said, medical emergency, so there's not really much time to get you up to the operating room, do a lot of anesthesia. We're just gonna kind of pump you full of drugs and slice open your fascial layer. All right, so if we look at skeletal muscle fibers, remember I said that they have a lot of different names, so we call them myofibers, muscle fibers, myocytes. Remember, myo means muscle, cyte means cell, so we can also call them muscle cells. They are multinucleated, meaning they have many, many nuclei per muscle cell. And they lie right underneath that cell membrane or that sarcolemma. Remember I said that sarcolemma is almost like the cell membrane of a muscle cell or a myofiber or a muscle fiber, myocyte, muscle cell. They all mean the same thing. So lots of nuclei per muscle cell that lies right underneath that sarcolemma. And it says that we could have 30 nuclei per millimeter of muscle tissue. And we'll come back to that here in a second. Says it's striated, long, and unbranched. So when we look at muscle tissue underneath the microscope, we'll see that it says it's striated, meaning that we'll be able to see these alternating light and dark patterns. And I'll just skip ahead a few slides. So here's what muscle tissue kind of looks like underneath the microscope. And you can see that there's alternating patterns of light and dark. And that's what I mean by striated. And we'll get to this in a second. Muscle tissue, skeletal muscle tissue can be very, very long. It can actually be over 20 centimeters long. So 20 centimeters is actually almost 8 inches. So if you look at your quadriceps, your muscle cells can be almost 8 inches long. So if we do the math, 30 nuclei per millimeter is actually 300 nuclei per centimeter times 20 centimeters is 6,000 nuclei per muscle cell. So why do we need so many nuclei per myocyte or per muscle cell? And the answer is really more efficiency. If we have a muscle cell that's, this long, we don't want the nucleus to be over here and to create a bunch of proteins that we have to transport all the way down to the other end of the cell. We want all of these nuclei so that we don't have to transport what we need to the other end of the cell. We want the nuclei to be really close to the area that we're going to need to produce the proteins, to produce whatever we need for skeletal muscle contraction. All right, sarcolemma, I said, was the kind of like the cell membrane of the muscle fiber. And we'll talk about the neuromuscular junction later on. The sarcoplasm, that's like the cytoplasm of the muscle cell. The sarcoplasmic reticulum I mentioned is like the endoplasmic reticulum of the muscle, and that's where calcium is going to be stored. So that sarcoplasmic reticulum is going to be up in orange. And like I said, that's going to be very important for calcium storage. Calcium plays a big role in skeletal muscle contraction. And I forgot to mention, mitochondria. Again, for nuclei, why is everything? Why do we have so many nuclei? Obviously, I mentioned to synthesize proteins so that we don't have to carry those proteins down almost 8 inches. But we know that mitochondria produces ATP, energy. So we have about 5,000 to 8,000 mitochondria per myocyte or per myofiber. And that's just efficiency. And what we'll see is if you're an athlete, let's say you run track, you might have more mitochondria than that, especially in your leg muscles. If you are a long distance swimmer, you might have more mitochondria in your arm muscles because you're going to use your arm muscles as a swimmer more than someone who doesn't swim. So it's all about efficiency and what your body needs are. So some of you might have more mitochondria because of your body demands than other people. You might have more organelles. And you can see here, mitochondria are kind of interspaced throughout the myocyte. All right. Again, many myofibrils. Not to be confused with myofibers. Remember, myofibers are your muscle cells, your myocytes. And remember, going back up to this slide, myofibers, and then if we remove one of the myofibers, then we get those myofibrils. So lots of myofibrils. Myofibrils are just a bunch of parallel structures that run down the myofibers. And they consist of these things called actin and myosin. And when we look at actin and myosin, we can see these little these purples. I guess we'll say that this is blue and that this one is purple. So the blue structure is actin, and the purple structure is myosin. So you can see on this slide that the blue actin is a little bit thinner than that purple myosin. So the blue is thin and the purple is thick. So we say that actin is thin and myosin is thick. And I mentioned when I went down to this slide, you can see this thing called a sarcomere. So a sarcomere is basically almost like a functional unit of skeletal muscle contraction. We'll get into what a sarcomere is a little bit later on. But basically, a sarcomere are units that connect end to end, and it says Z line to Z line. And I'll show you a picture of this here in a few slides. But they make up muscle fibers or myofibrils. They're segments of a muscle fiber. And like I said, they're the functional unit of skeletal muscle contraction. And so, don't worry about this for right now. We'll look at this here in a few slides. We also have these things called transverse tubules. That's what is kind of like these yellow things. Remember sarcoplasmic reticulum, that's the orange things. That's what stores calcium. But we also have these transverse tubules. We also call them T-tubules. And that's going to be what transmits those action potentials or those impulses into the cell via the sarcoplasmic reticulum for actual skeletal muscle contraction. And Remember, if I don't know if you talked about this in Bio 1000 or entry level class, that we have that nerve impulse that comes down that stimulates or that has that action potential that stimulates the muscle cell to contract. And what happens is that the sarcoplasmic reticulum gets stimulated and the transverse tubules get stimulated, and we release a bunch of calcium, and that the transverse tubules actually kind of disperses that action potential. So when we look at skeletal muscle and cardiac muscle, those transverse tubules are actually very well developed in skeletal muscle and cardiac muscle, but it's not so well developed in smooth muscle. So transverse tubules, sarcoplasmic reticulum, very well developed in skeletal muscle, well developed in cardiac muscle, not so well developed in smooth muscle. All right, so I mentioned that we have this striation pattern of skeletal muscle, and that's because of the actin and myosin. And we looked at that picture a little bit here. It's because of that actin and myosin of that organization that gives us that striation pattern. All right, and I briefly mentioned the sarcomere. That's that functional unit of skeletal muscle contraction. And that sarcomere, and if we go back to this slide, you can see how some portions we only have actin, some portions we only have myosin, some portions we have actin and myosin overlap. So when we look at it under the microscope, some portions will appear dark purple, some portions will appear only blue, some portions will appear a mixture of blue and purple, whatever blue and purple looks like when it's mixed. And so we can kind of divide this section into I bands and A bands and H zones and Z lines and M lines. And we're not really going to go over this portion in class. We will for lab. So I'm not really going to go into this in class, but we will talk about the sarcomere. So remember that the purple is going to be our thick filament or myosin, and that blue structure is going to be our thin filament or actin. And you can see that we have, this little squiggly line, which is our blue line, which is just actin. It's just our thin filament. So because it kind of looks like a Z, we actually call that a Z line. And then again, over here, we have a little squiggly line, again, another Z line. So from Z line to Z line, that's what we call a sarcomere. All right, and then we have another area, kind of like divided here, of just our thick filament myosin. So here, if we just have our myosin, we actually would call that our H zone. And then in other areas, so here we would have just actin. So we would just have blue stuff. So that's actually our I band. And then, here, if we have, let's say, some actin and myosin that are overlapping a little bit, that's our A band. So this is how we get all of those A bands and H zones that I was talking about in the previous slide. that we're really not going to go over. And then if you draw a line down through the center of just our only myosin portion, that's going to be our M line. M is for myosin. So remember, if we go back to this slide, we have two filaments, our actin and our myosin. So if we look at our myofibril, it's basically made up of actin and myosin. All right, so that's how we get all of our A lines and our M lines and stuff like that, our A bands. So we're not really going to focus too much on this for class. We'll go over it in lab. But what you do need to know for the class portion, sarcomere, Z line to Z line, or actin only to actin only. All right, so for here, sarcomere, this is our Z line. This is our Z line. You know, that little squiggly line. So our sarcomere goes from actin only, or the middle of our actin only, to the middle of our actin only. That's the functional unit of skeletal muscle contraction. Actin's our thin filament, myosin is our thick filament. All right, so kind of to recap, thin filament, actin. Thick filament, myosin. Now, when we look at actin and myosin, actin really is kind of intertwined with two other molecules, troponin and tropomyosin. And at rest, troponin and tropomyosin are covering up the binding sites of actin. So at rest, actin and myosin cannot bind. So at rest, these myosin heads will not be able to bind to actin. So the thick filament and the thin filament cannot bind together. So that's why it says associated with troponin and tropomyosin, which prevents cross-bridge formation when muscle is not contracting. So at rest. And cross-bridge just means when they are bound together. Bound together. All right, your thick filament, remember, is myosin. We have these myosin heads, and they're kind of like a double-headed. There's like two heads on each head. So the two heads of myosin will eventually bind together or form a cross bridge with actin once those binding sites of actin are exposed. Once we can get the troponin and tropomyosin out of the way, those binding sites on actin will be exposed, and then myosin can bind to actin. And we'll go over that process later on. All right, so contraction of a skeletal muscle fiber, obviously it's the shortening of a skeletal muscle fiber as it pulls on its attachment points. And it requires a lot of different chemical reactions and cellular components. But what happens, it's a movement within the myofibrils And what happens is those actin and myosin filaments slide past each other and shortening the sarcomeres. So remember, those Z lines to Z lines get shorter. And what we'll see is that those myosin heads will bind to actin. And let me go back a slide. So eventually those myosin heads will bind to actin, and those myosin heads will flip inwards, and actin will actually get pulled closer to each other. So actin is what gets shortened. Myosin does not get shortened, but actin gets shortened. So that's why we say the sarcomere gets shortened, because remember, sarcomeres are actin, not myosin. Sarcomere is actin. Sarcomere is actin. Z line to Z line. All right, so now we're going to switch topics a little bit and talk about the neuromuscular junction, or the myoneural junction. And basically, we have this thing called a synapse. And a synapse is basically when a nerve connects to another nerve. A nerve connects to another nerve connects to a gland. A nerve connects to a muscle. And in this case, a synapse is when we have a functional connection between a neuron and a skeletal muscle fiber. And also, neurotransmitters are going to be released. So we have to have a neuron or a motor neuron release these neurotransmitters, and in order for that skeletal muscle to contract. So motor neuron. So remember, we briefly talked about this earlier on. We have sensory neurons and we have motor neurons. Sensory neurons, you put your hand on the hot stove, sensory neurons will tell your brain that the stove is 325 degrees, and it's going to get processed in your hypothalamus. And then you're going to have motor neurons, and your brain's going to say, oh my gosh, the stove is really, really hot. And then you're going to have motor neurons that remove your hand from the hot stove, or that provide some sort of action. So motor neurons perform an action. So remember the difference between sensory neurons and motor neurons. So a lot of times I'll say nerves and neurons. So they're essentially the same thing, neurons and nerves. All right, so just to kind of go over this slide a little bit, we have mitochondria, because if you, I'll get to that in a second. So we have, first we have these vesicles. These vesicles contain neurotransmitters called acetylcholine, and eventually, once the action potential or that electrical impulse comes down the length of the axon and stimulates this neuron, these vesicles are going to bind to this axon terminal, and these vesicles are going to bind to this neuron cell membrane, and the vesicles bind to the cell membrane and dump out their contents into what's called the synaptic cleft. So this space right here is called the synaptic cleft. You can see it right here, synaptic cleft. Now, if you remember back from chapter 2 or 3 or whatever, when vesicles release their components, into the synaptic cleft, or when vesicles release their components, that's an active process which requires energy in the form of ATP. So that requires mitochondria in order to make ATP, and we learned that in chapter 4, creating energy, glycolysis, citric acid cycle, electron transport chain. So that's why we need ATP. Now, one thing that I did not mention is in order for these vesicles to release their contents into the synaptic cleft, we're going to have calcium in order to do so. So calcium is going to need to be taken up into this axon terminal in order for the vesicles to be able to fuse to that axon terminal. All right, so the vesicles have now dumped their contents into the synaptic cleft. These acetylcholine molecules are going to go down into these little receptors down here, and they're going to bind to these, for now we will call them acetylcholine receptors. We'll learn later on that they're not really acetylcholine receptors, but for the purposes of now, we'll call them acetylcholine receptors. So they bind to acetylcholine receptors, and then that's the first process of skeletal muscle contraction. So here you can see that so we have remember we have our neuron, that we have our, dendrites, cell body, and then we have our axon. Remember, neurons can have thousands of dendrites, then a cell body, and it can only have one axon. But that axon can branch off, they're called collaterals. So this axon actually branches off into 4 different collaterals. And then we blew up the picture here, and that's what we see. And we have what's also called a motor end plate. So this motor end plate is basically a folded portion of that sarcolemma, of that skeletal muscle. It's basically an epithelial layer of that skeletal muscle tissue. All right, moving on. All right, so we talked about the motor neuron. That's a neuron or a nerve that controls the skeletal muscle fiber. We talked about those synaptic vesicles that contain neurotransmitters. And in the case of skeletal muscle contraction, that neurotransmitter is going to be acetylcholine. We actually have about 100 different neurotransmitters, and they all do different functions. But for skeletal muscle contraction, it's going to be acetylcholine. So neurotransmitters, that's going to be those chemicals released by the motor neuron to deliver whatever message we're trying to deliver. In this case, we're trying to initiate skeletal muscle contraction. We have that synaptic cleft, that space between that motor neuron and that motor end plate. So that motor end plate, remember, is that specialized folded portion of that skeletal muscle fiber, that sarcolemma, where the neurotransmitter binds to those receptor sites, those acetylcholine receptors. Now, generally, muscle fibers only have one motor end plate, kind of per axon. So here you can see we have one axon and one motor end plate. But a single motor neuron can innervate many different muscle fibers in a group. And it says a signal from one motor neuron can potentially stimulate multiple fibers in a group to contract, generating more forces. So here it says one neuron or one motor neuron stimulates one motor end plate, but a single motor neuron can innervate many different muscle fibers. So here we have, that's why our axon branches. 1, 2, 3, 4. Now this example shows 4, but in reality, we might have 100, we might have 1,000. So when we contract a muscle, we don't contract just one myofibril or one myofiber. We actually contract the muscle as a group. We contract the whole bicep brachii. We don't contract 2 or 3 different myofibers within the biceps brachii muscle. We contract the whole muscle as a group, or the whole muscle. That's just what these statements say. So we have the neuron that can stimulate kind of like the whole muscle. And the other definition is a motor unit. So a motor unit is that motor neuron and the muscle fiber that it innervates. So basically a motor unit is going to be that motor neuron and the muscle fiber that it innervates. All right, so acetylcholine is a neurotransmitter for skeletal muscle contraction. And when we talk about skeletal muscle contraction, we can talk about it from many different perspectives. We can look at it from a nervous system perspective. We can look at it from a skeletal muscle perspective. So here we're going to look at it from a nervous system perspective. We actually kind of already mentioned this up here. All right, so acetylcholine is a neurotransmitter. Acetylcholine is actually synthesized in the cytoplasm of the motor neuron. So the cytoplasm of the motor neuron is in the cell body. So acetylcholine is actually synthesized here, and then it's transported down the axon, and then it's going to be stored in these vesicles in that distal end of the axon until they are ready to be released. So they're stored in those synaptic vesicles near the distal end of the axon. All right, so then at some point, that nerve is going to be stimulated. Say, all right, we're ready to fire. It's time to contract that skeletal muscle. So that nerve impulse releases or causes the release of acetylcholine from that axon terminal of that motor neuron, specifically those synaptic vesicles. So those synaptic vesicles releases those synaptic vesicles fuse to the cell membrane in the presence of calcium, and dumps their acetylcholine in that synaptic cleft. Now remember, it's exocytosis, so it's an active process which requires ATP. So then that acetylcholine diffuses across the synaptic cleft at the neuromuscular junction. Acetylcholine binds to those acetylcholine receptors on that motor end plate. Remember, that's the sarcolemma, that folded portion. Now, acetylcholine then causes a change in that membrane permeability. So sodium's going to influx and potassium efflux, which generates a muscle impulse or an action potential. So this influx just means comes into the cell, and efflux means goes out of the cell. So it says acetylcholine causes changes in membrane permeability so that sodium rushes into the cell and potassium rushes out of the cell. And we're actually going to save this statement for next chapter. We're going to spend not a lot of time. We're going to spend some time talking about this statement next chapter. All right, so then that impulse causes the release of calcium from that sarcoplasmic reticulum. Remember that orange structure. This orange structure right here. And remember, these are the transverse tubules, that yellowish structure. So that impulse causes a release of calcium from that sarcoplasmic reticulum, and it's going to diffuse into that sarcoplasm, kind of like the cytoplasm of the muscle, and bind to troponin. Remember, troponin is actually one of those molecules that is surrounding or kind of blocking one of those binding sites on actin. So calcium binds to troponin, which we'll get into later on when we talk about skeletal muscle contraction from a skeletal muscle perspective. Right now we're talking from a nervous system perspective. All right, so calcium binds to troponin. Got to remember that. All right, then the muscle fiber membrane is stimulated, and that muscle impulse travels deep into the muscle fiber through those transverse tubules, those yellow structures. Now you're going to have to know these steps in order. And as we go through what happens from a skeletal muscle perspective, you'll be able to understand this process even more. All right, so botulism results from an ingestion of a toxin produced by a type of bacterium. Along with other symptoms, it causes a type of paralysis in which the muscles cannot contract. Why does this paralysis occur? So the toxin promotes the release of acetylcholine. The toxin prevents the breakdown of acetylcholine. The toxin prevents the release of acetylcholine, or the toxin binds to acetylcholine receptors at the neuromuscular junction. All right, so think about what acetylcholine does. Acetylcholine causes skeletal muscle contraction. So paralysis means you can't contract your skeletal muscles. So the toxin promotes the release of acetylcholine. If we're releasing acetylcholine, we're going to have skeletal muscle contraction. No. If it prevents the breakdown of acetylcholine, that means we still have acetylcholine. So we're still going to have skeletal muscle contraction. All right, so the toxin prevents the release of acetylcholine. So that means we don't have acetylcholine in that synaptic cleft. So that's a viable answer. The toxin binds to acetylcholine receptors at the neuromuscular junction. If the toxin binds to acetylcholine, therefore acetylcholine can't bind to those acetylcholine receptors. Therefore, we don't have skeletal muscle contraction. So that's a viable answer as well. So how do we kind of go through that? How do we rationalize that? So let's talk about A and B first. So if you're paralyzed, that means acetylcholine isn't working. These A and B means that you have too much. These two statements so for A and B, if you're paralyzed, that means that acetylcholine isn't working. All right, so for C and D, so these two statements mean, okay, for answer D, now let me go back. Let me look at my answers for a second. Okay, if you're paralyzed, that means acetylcholine really isn't working. So for answers B and D, these two statements mean that you have too much acetylcholine. You would see cramps, increased salivation, lacrimation, tearing, muscle weakness, paralysis, severe muscle fasciculation, twitching, diarrhea, blurry vision. This is called cholinergic crisis. We actually talked about this in chapter 4. It's what happens with when you spray Roundup and your pets actually accidentally ingest those pesticides. Answer D. This is saying that botulism would prevent acetylcholine from binding to those receptor sites. If this were the case, you would be paralyzed, but you would also experience cholinergic crisis. So we're going to throw out answer D, leaving us with answer C as the most viable answer. So botulism actually prevents the release of acetylcholine. All right. So now we're going to talk about what happens with skeletal muscle from a muscular system perspective. And some of these will be kind of repeated from a nervous system perspective. So that impulse arrives from the neuron at that synapse. We know that acetylcholine is going to be released from that axon of the motor neuron. We know that the impulse then travels through those transverse tubules. Now, the muscle impulse then reaches the sarcoplasmic reticulum and calcium is released. This is so far kind of like the same from the nervous system perspective. Calcium floods that sarcoplasm So far the same. Calcium then binds to troponin molecules. So far that's the same. And it leaves those active sites exposed. And then those myosin heads can bind to those exposed active sites on actin, forming those cross bridges. Remember, cross bridges are when myosin binds to actin. Then those myosin heads actually flip in on itself. Then those thin filaments, actin, are pulled over the thick filaments, and then the muscle fiber shortens and contracts. And I'm going to pause the video here because there's some pieces of information missing. So I'm going to pause and actually rework this slide. All right, so here's what I wanted to change. So we know that calcium floods the sarcoplasm and that it binds to troponin. Then, because of that, troponin is going to alter its shape. Once troponin alters shape, that allows tropomyosin to move a little bit. Once tropomyosin kind of moves out of the way, this leaves those active binding sites on actin exposed. And now those myosin heads can bind to those exposed active sites on actin. They form those cross bridges. Then myosin has now bound to actin. Those myosin heads flip in. So those thin filaments, actin, are pulled over the thick filaments, myosin. The muscle fiber shortens and contracts. So we'll do this. Are pulled over the thick filaments, myosin, muscle fiber shortens and contracts. All right, so excitation, contraction, coupling. So basically, this is a connection between the muscle fiber stimulation and muscle contraction. So a lot of this is just a rehash of what we've already talked about. So during muscle relaxation, so when muscle is not contracted, calcium is going to be stored in that sarcoplasmic reticulum. That troponin and the tropomyosin is going to be covering up those binding sites on actin. And then when the muscle is stimulated, the sarcoplasmic reticulum releases calcium into that cytosol or into that sarcoplasm. And that calcium binds to troponin to change the shape of troponin. And then the troponin, because the troponin changes shape, tropomyosin will change shape. Then the binding sites of actin are now exposed. Myosin then binds to actin. That cross bridge is now formed. Then so we have that myosin attaches to actin. We formed that cross bridge. Myosin pulls actin towards the center of the sarcomere. So it pulls that thin filament, we'll call that actin, towards the center of the sarcomere. That pulling of that actin molecule actually requires energy. So ATP is broken down into ADP plus phosphate. We actually call that a power stroke when the myosin heads flip in on each other. That's called a power stroke. So because we used ATP in that process, we now need some more ATP. So we have done a power stroke, which we've used ATP. Then that link between actin and myosin breaks off. So the myosin actually, those myosin heads flip back outwards. And as long as we have calcium and enough energy, those binding sites on myosin will bind, or and as long as actin still has those binding sites exposed, we can reestablish that link between myosin and actin, and maybe create another contraction. So that linkage between actin and myosin break up. We use more energy, and then that cross bridge goes back to its original position. Those myosin heads flip back, ready to bind to another binding site on actin, as long as we have calcium and energy available. So looking at this from a picture standpoint, here on our actin, we have troponin and tropomyosin blocking the actin, blocking those binding sites on actin. So once calcium gets released from that sarcoplasm, calcium binds to troponin, troponin alters shape, tropomyosin moves out of the way, and those binding sites are now exposed. So those exposed binding sites on actin are now exposed. Myosin binds to actin, forming cross bridges. Myosin binds to actin. Now, we use ATP in order to pull actin. Myosin heads flip in and pull actin closer to that sarcomere. We call that a power stroke. Myosin heads flip in and we use ATP. Then that connection is broken. See, that connection is broken there. Those myosin heads return to its original position, and it's ready for another contraction as long as we have enough calcium to keep those binding sites exposed to generate another contraction, or it'll go back to a relaxed state. And if we go back to a relaxed state, then the troponin and tropomyosin will cover up those binding sites. All right, so during a muscle contraction, why is ATP required? All right, to break down glycogen into glucose for fuel. No. Well, glycogen is stored glucose. ATP is required. ATP is typically not required to break down. Is typically not required to break down something. ATP is required to put things together. To stimulate enzyme activity. So here we haven't even talked about any enzymes that are occurring. To synthesize creatine phosphate. We haven't even talked about creatine phosphate. To allow myofilament movement, myofilaments, actin, myosin, ATP is required to move actin. So that's going to be our answer. All right. We also can look at what's happening. We talked about from a nervous system perspective. We talked about from a muscular system perspective. We can actually look at what's happening from a microscopic perspective. What's happening under the microscope? We have our sarcomeres, our A zones, our H zones, our Z lines. So basically, we know that the sarcomere is shortening because actin is moving closer together. Actin slides across myosin. So we know that our sarcomere is shortening because actin is getting pulled closer together. So we can see that our H zones and our I bands are getting closer together. Our Z lines getting closer together. Even though our thin and thick filaments aren't changing in length, but we do have those overlapping between an overlapping of our thick and thin filaments. We're not going to know, you don't need to know the sliding filament model for classroom purposes, but you can see what's happening to the sarcomere as skeletal muscle contracts. So here's our Z line. And as our skeletal muscle is contracting, you can see that the sarcomere is getting shorter. All right, in a laboratory that studies muscle function, a group of rats are bred that, due to a genetic defect, a non-functional version of troponin is produced. As a result, the rats are paralyzed. What explains the impact of this defect on muscle? So a non-functional version of troponin. So think about what happens with troponin. Troponin, which is exposed when calcium binds to actin. So we can throw that one out because calcium binds to troponin. Calcium does not bind to actin. B, troponin can't bind calcium ions and thus will not change shape nor cause tropomyosin to move away from the binding sites on the thin filament. All right, so far that's a good answer. Troponin, which should alter shape when calcium ions bind to tropomyosin. Calcium does not bind to tropomyosin. Troponin, when calcium ions bind to tropomyosin. No, calcium binds to troponin. So that's going to be our answer. All right, in a laboratory that studies muscle function, a group of rats are bred due to a genetic defect, have myosin heads that bind ADP irreversibly. As a result, the rats are paralyzed. What explains the impact of this defect on muscle? So let's go back and look at ADP. So let's go back to this one. So here's where I talked about ADP. So we have calcium has been bound, we formed a cross bridge, and we've created a power stroke. But we have not released that cross bridge. So that's where we are. All right, so ATP is unable to bind to the myosin heads, preventing the actin binding sites of the thick filament from being refilled. So there's a lot wrong with this. So preventing the actin binding sites of the thick filament. So one, actin is on the thin filament, not the thick filament. So we know that's wrong. Because ADP is permanently bound, the myosin heads are unable to attach to the actin binding sites on the thin filaments. So we go back up here. We've already created a cross bridge. So that's what this statement is saying, that we can't form a cross bridge. We've already formed a cross bridge with ADP. We've already done a power stroke. All right, because ADP is permanently bound, the myosin heads will be irreversibly cross-bridged with the actin binding sites on the thin filament. So here we have formed a cross bridge, we've done a power stroke, and now we're stuck here. We are permanently cross-bridged. We can't relax. We are permanently contracted. So far, that's our answer. ATP is unable to bind to the myosin heads, causing the thin filament to be constantly pulled in the power stroke of contraction. So here, we've already bound ATP. See, we bound ATP. We're stuck here. So C is going to be your answer. We've actually are in a permanent state of contraction. We've done our power stroke and we're stuck in a permanently contracted state. All right, so what happens with that acetylcholine? We have done our skeletal muscle contraction, and now we have to get rid of that acetylcholine. We have an enzyme called acetylcholine esterase, and that's what decomposes or breaks down acetylcholine. We have to get rid of acetylcholine in that synapse, and acetylcholine esterase is what breaks down or gets rid of acetylcholine in that synapse. And that's what stops that muscle impulse from continuing. And what happens is that once we get that acetylcholine esterase, we stop that stimulus to the sarcolemma. The muscle fiber membrane ceases to continue that impulse. Calcium in that muscle fiber moves back into that sarcoplasmic reticulum. The troponin tropomyosin complex covers up those binding sites on actin. Myosin and actin binding sites are now prevented from binding together, and then your muscle fiber then relaxes. All right, curare. It's a poison that binds acetylcholine receptors, or binds to acetylcholine receptors at the motor end plate. This prevents acetylcholine from binding to it. It's called competitive agonist. What is the result of curare poisoning? So curare binds to those acetylcholine receptors and prevents acetylcholine from binding to it. So what's the result of curare poisoning? Lack of neurotransmitter release by the motor neuron. Well, we still have acetylcholine getting released, but curare is binding to that acetylcholine receptor. So answer A is going to be out. Rapid breakdown of acetylcholine by acetylcholine esterase. No. Inability of the muscle fiber to respond to nervous system stimulation. that would be correct because curare is blocking acetylcholine from binding to those receptors. So we can't contract skeletal muscles because acetylcholine is not binding to those acetylcholine receptors, thereby not doing steps, 4 through 27 of skeletal muscle contraction. Increased acetylcholine production. No, we're producing acetylcholine just like we should. It's just not getting to those receptors. All right, so here's a summary slide of muscle fiber, of muscle contraction. We've already been over this. Same thing for muscle fiber relaxation with acetylcholine. All right, so ATP. We talked about ATP earlier in a previous chapter. And when we break down ATP, this is our adenine molecule plus our ribose sugar plus our 3 phosphates. We're actually going to break this bond right here. And when we break that bond, we're going to go ATP to ADP plus our 3rd phosphate plus some energy. And when we break that bond, we actually generate 7.3 kilocalories per mole of energy. And that's a decent size of amount of energy. But, and that's going to be our first source of energy for muscle contraction. But we only have so much ATP available. It's not like, everything goes back to homeostasis. We don't want too much ATP. We don't want too little ATP. We only want just enough of what our body needs are at the time. Now, some of you might ask, well, why can't we break this 2nd phosphate group or this 3rd phosphate group? So here I said we get 7.3. If we break off this 2nd phosphate group, we actually only get 3.3 kilocalories per mole of energy. And if we break off that 3rd phosphate group, we actually only get 1.6 kilocalories per mole of energy. So it's not really advantageous to the body to break off this 2nd or 3rd phosphate group. We get more energy from that 3rd phosphate group. And I mentioned in a previous chapter that we can break off this 3rd phosphate group and then we can add it back on. It's called phosphorylation. Then we can break it off again. Then we can add it back on again. Then we can break it off and get another 7.3. We can add it back on, break it off again, add it back on, break it off, and we can still get that 7.3 kilocalories per mole of energy every time we break it off. But what we have is another way to create energy. We have this thing called creatine phosphate. And for those of you who use protein powder or do weightlifting, you've probably heard of creatine phosphate. It's a supplement that you can take as kind of like an energy source. Basically, it will supply energy to synthesize ATP from ADP and that inorganic phosphate. So we could actually make this equation a double-headed arrow. Remember that the body, most reactions in the body are a reversible reaction depending on what those body needs are at the time. And when we use creatine phosphate, what we will do, let's say that, we're not going to need to know the chemical structure of creatine phosphate, but when we break off that phosphate group of creatine phosphate and add it back on into this equation, we're actually going to get 10.3 kilocalories per mole of energy, which is going to be more than that 7.3 kilocalories per mole. So, and also what we find is that it's actually more abundant, 4 to 6 times more abundant than the ATP in our body. The downside is that one, that I told you that we only store a little bit of ATP, and two, when we look at ATP and creatine phosphate, we really don't have that much energy available. We can only use that for about 10 seconds of intense muscle contraction. So when we look at this picture here, if we have I'll start here. When we have low ATP storage, or when we have low ATP in our body, we'll break off this creatine phosphate, or we'll break off that phosphate from that creatine phosphate to add, to get 10.3 kilocalories per mole to phosphorylate that ADP into ATP to use for energy. And then when our ATP stores are high, we'll break off that third phosphate of ATP to get our 7.3 kilocalories per mole to add back on to creatine phosphate. And then we can just kind of alternate back and forth between using creatine phosphate versus ATP to break down either creatine phosphate or ATP, depending on whatever we have more of. All right, remember cellular respiration, glycolysis, citric acid cycle, electron transport chain. So we have cellular respiration. However, and that can be used for long periods of muscle contraction. Remember that we have, we break down glucose to produce ATP in glycolysis, citric acid cycle, and all that stuff, electron transport chain. And remember that we have stored glucose in our liver cells and our skeletal muscle cells. And let's just write liver and skeletal, S-K-E-L-E, skeletal muscle cells. So we have that glycogen as a backup when we run out of glucose. Glycogen is just stored glucose. So we have that as a backup source of energy as well. However, sometimes we're going to run out of energy. So just as a recap from chapter 4, we have anaerobic phases of cellular respirations. Remember, anaerobic is without oxygen. That's glycolysis. It occurs in the cytoplasm, and it produces very little ATP, both very little direct ATP and very little indirect ATP. And then we also have aerobic respiration. It's oxygen. That's our pyruvate oxidation, citric acid cycle, and electron transport chain. Occurs in the mitochondria. Produces the most direct ATP and indirect ATP. Remember, indirect ATP are those NADHs and those FADH2s. All right. Myoglobin. It stores the oxygen in the muscles. Remember, we have hemoglobin. And basically myoglobin is just hemoglobin inside the muscle tissue. Myoglobin is the protein that contains that heme molecule. Heme is also inside hemoglobin. That's what carries oxygen. Myoglobin is responsible for that reddish brown color of skeletal muscles. Remember, so just the picture that we've seen before, glucose is in the cytoplasm, gets converted to pyruvic acid, which moves into the mitochondria for the citric acid cycle, electron transport chain, hopefully produces a bunch of ATPs. Now, from here, what we'll see on the next slide, what we're going to talk about is this thing called lactic acid. All right, so when we exercise, let me start over. When we're at rest or when we do moderate exercise, typically the respiratory system, the cardiovascular systems supply enough oxygen to support both anaerobic and aerobic respiration. So all of cellular metabolism, glycolysis, citric acid cycle, electron transport chain, all of that. However, if we are doing strenuous activity, we find that the cardiovascular system, the respiratory system, does not provide enough oxygen for skeletal muscles to work properly. We only provide enough oxygen to support those muscles for about a minute or two. So we call this anaerobic threshold or lactic acid threshold. And what happens is we have this shift in metabolism from an aerobic metabolism to anaerobic metabolism. Like I said, we really only see this during periods of strenuous muscle activity. So go back and remember what happens during anaerobic versus aerobic respiration. So here we're talking about anaerobic respiration, glycolysis. And remember that I said glucose goes to pyruvic acid, and then typically pyruvic acid, acetyl-CoA goes to the citric acid cycle. But instead, pyruvic acid, we can actually convert pyruvic acid into lactic acid, which is what's going to happen. So because we are going to switch over to anaerobic metabolism, we're going to see that glycolysis still happens. Glucose is still going to get converted into pyruvic acid, but instead of pyruvic acid normally getting converted into acetyl-CoA, because we don't have that oxygen supply, pyruvic acid is going to get converted into lactic acid. And lactic acid is going to go to the bloodstream. It's going to then go to the liver to create more glucose. The cycle is going to repeat. So we're going to stay in an oxygen-deprived environment, anaerobic metabolism. So we're going to kind of go through glycolysis over and over and over again. So here, remember, we have glycogen, which is just stored glucose, and that glucose gets converted into pyruvic acid, and instead of going to acetyl-CoA and following aerobic metabolism, citric acid cycle, that pyruvic acid is going to get converted into lactic acid, goes to the liver to get converted into glucose, and the cycle is going to repeat itself. All right, so now most of that oxygen from that strenuous exercise is actually used to produce ATP for muscle contraction, not for converting lactic acid into glucose. So remember, in order to contract skeletal muscles, we have to break down ATP. We saw that in that previous several slides ago when we do the power stroke. So we use that oxygen to create ATP for those power strokes for muscle contraction, not converting lactic acid into glucose. So when we are done with that strenuous exercise, we're left with what's called oxygen debt. And basically that oxygen debt is that amount of oxygen needed by liver cells to kind of convert that lactic acid back to glucose or glycogen, and also to restore that muscle ATP and the creatine phosphate back to its original concentrations. And remember that oxygen develops because the respiratory system and circulatory systems are unable to supply enough oxygen for skeletal muscles to work strenuously for a minute or two. And if you're working out, hardcore running for an hour, you're going to be left with severe oxygen debt. So our ATP levels, your creatine phosphate levels, your oxygen levels, and your glucose levels all have to be returned to normal. And it can actually take several hours to get those levels back to normal. And if you get cramps, it's due to low ATP, low levels of ATP. Now, I will say that this statement right here develops because the respiratory and circulatory systems are unable to supply enough oxygen to skeletal muscles to work strenuously for a minute or two. That's almost a lie. And we'll learn about this next semester in AMP2. Your cardiovascular system, when we look at the amount of oxygen, that when we breathe in the oxygen, we can measure your bloodstream and like your arteries and determine how much oxygen is in your arteries. And then we can look at your blood that returns to your heart and see how much oxygen is still bound to your hemoglobin. And let's say that you have 100% oxygen bound to your hemoglobin in the artery side, but when your body returns the blood back to your heart, actually you have about 75% oxygen still bound to your hemoglobin. So we have the oxygen available. Your body just doesn't dump it off at the cellular level. So we have the oxygen available. Your body just doesn't dump it off at the cellular level. All right, so with some muscle issues, muscle fatigue. If you run a marathon, you might be inability to contract muscles. You see the marathon runners at the end of the marathon, they fall down. And it's because of decreased blood flow, ion imbalances across that sarcolemma, across that muscular cell membrane. I'm not sure about a loss of desire to continue exercise, but big one, accumulation of lactic acid. Remember that lactic acid builds up and builds up. You are altering your pH. Lactic acid is an acid. You have the accumulation of hydrogen ions. You are adjusting your pH. You are out of homeostasis. Muscle cramps. We've all had muscle cramps, I'm sure. Involuntary muscle contraction. Those are your Charlie horses. It's caused by changes in electrolyte concentrations between those intracellular and extracellular fluids in the muscle areas. All right, why is it that athletes have developed better endurance than non-athletes, or why don't they develop muscle fatigue quicker? Obviously, it's due to training, but what is it about the training? What is occurring physiologically? All right, athletes can use ATP more efficiently. Athletes can convert glucose to lactic acid more quickly. Training develops a better cardiovascular system, and athletes have a better blood supply and more mitochondria. Athletes can tolerate higher concentrations of carbon dioxide. All right, so athletes can use ATP more efficiently. So if you can figure out a way to use ATP more efficiently, then you can probably become a billionaire, multi-billionaire. All right, so we're going to get rid of that one. All right, athletes can convert glucose to lactic acid more quickly. We actually don't want to convert glucose to lactic acid more quickly because that's just going to alter your pH more. One of the most common causes of muscle fatigue is the accumulation of lactic acid. This question is asking about oxygen debt. This is a trick answer. The answer is saying to convert glucose to lactic acid. This already happens in glycolysis through pyruvic acid. If we can find a better way to convert lactic acid to glucose, then this would be a viable answer. So we'll cross off B. We'll look at D. Athletes do produce more carbon dioxide than non-athletes, but that doesn't mean they can tolerate higher concentrations of carbon dioxide. So actually, if you look at athletes and non-athletes or whatever, when we're exercising, you're breathing faster. So your cells are actually producing more carbon dioxide as a result of cellular respiration, because if you go back to the equation that we learned about in chapter 4, we produce carbon dioxide as a result of cellular respiration. But because of your increased respiratory rate and increased heart rate, your blood is moving faster and you're breathing out that carbon dioxide faster. So you really don't have a higher concentration of carbon dioxide in your bloodstream. So we're going to throw out answer D. Answer C is actually the correct answer. Athletes have developed a better cardiovascular system because of exercise. The body is great at adapting because their bodies have increased activity. They have more mitochondria. I mentioned that earlier in the chapter or earlier in the lecture. Their cardiovascular system, they have developed blood vessels that are really intertwined in their muscles to help remove waste products. So in this case, C will be our answer. All right, so heat. Remember that heat is a byproduct of cellular respiration. We either produce energy in the form of heat, or the other 40% will be released as chemical energy. So muscle contractions are a major source of body heat, helps maintain body temperature. You get cold, you shiver, that heat goes through your core, and hopefully you warm up a little bit. All right, so, all right, threshold stimulus. We'll talk about this next chapter a lot, but this is the minimum stimulus required to contract a muscle fiber. And I use the analogy of like your brake lights. If you barely tap on your brakes, your brake lights will come on. If you slam on your brakes, your brake lights will come on. If you whatever this threshold is, if you reach that threshold, your muscle fiber will contract. If you, as long as you reach that threshold, your muscle fiber will contract. If you reach that threshold, that action potential will be generated. That calcium will be released. Your muscle fiber will generate that action impulse. All right, so remember that a motor unit is that motor neuron and that muscle fiber that it innervates. It's that axon terminal, that synaptic cleft, that sarcolemma, that folded sarcolemma, and that whole muscle fiber. Now, recruitment is an increase in that number of motor units. Remember that one axon can divide into multiple collaterals to produce more force. You know, we want that whole muscle to be stimulated and not just one myocyte. So, you know, you contract your whole bicep as opposed to one myocyte. And we use more stimulation. We increase that intensity of stimulation to recruit more motor units in hopes that all motor units are activated. All right, we have basically two different types of contractions, isotonic and isometric contractions. Isotonic contractions are basically when you are actively flexing and extending when you have weights in your hand. We can break down isotonic contractions into concentric and eccentric contractions. So concentric contractions is when you are flexing your muscle, so you're shortening that contraction, such as like an abdominal crunch. Eccentric is when you are extending that contraction, and isometric contraction is when you're still working out that muscle, but you're not using weights, like holding a plank. That would be an example for an isometric. You're still working out that muscle. Remember your definitions. You're going to have some examples on your quiz and your exam about some of the definitions like flexion, extension, anterior, forearm, insertion, posterior, tibia, origin, femur, abduction, adduction. All right, so muscles. We have different types of skeletal muscles based on their contraction speed. We have slow twitch muscles, fast twitch muscles. How do they produce ATP? Do they produce it oxidatively or glycolytically? We're actually not going to get into how they produce ATP. We're not going to go into oxidative versus glycolytically. But we have three different types of muscle fibers. We have slow twitch muscles. Those are your type 1 oxidative muscles. We have fast twitch muscles. We can classify those as type 2A, which are fast twitch oxidative muscles, type 2B, which are fast twitch type 2B glycolytic muscles. And those muscles, for slow twitch muscles, those are, your type 1 oxidative muscles. Those are going to be slow twitch fibers that are very resistant to fatigue. So if they're very resistant to fatigue, they're going to have a lot of mitochondria that that provide you with a lot of ATP. They're going to have a good blood supply. They're going to have a lot of myoglobin, so they're going to be provided with a lot of oxygen. However, they're going to be slow to contract or slower to contract, hence the name slow twitch. Now, these slow twitch fibers, they're going to be used for longer use activities. So for like distance swimming or distance running, long distance activities. So like your back muscles or leg muscles for long distance running. So for your fast twitch muscles that are oxidative, like your type 2A muscles, they're going to contract quickly. They're going to have a little bit amount, an intermediate amount of myoglobin, an intermediate amount of blood supply. But we'll jump over to the fast twitch type 2B that are glycolytic. They're going to contain a lot of glycogen. So they're going to be able to convert that glycogen back to glucose. They have a limited blood supply. So they're going to have fewer mitochondria. They're going to be more susceptible to fatigue. And they're going to be larger in diameter. So just with these, just kind of memorize the underlined stuff. There's not going to be really any hard questions about these muscles, so just kind of memorize what's underlined. Here's just a summary slide. You really don't need to know that chart. All right, some disuse of muscles. Hypertrophy, it's not really a disuse. Hypertrophy is just muscle enlargement. You lift weights and you gain muscle mass. We have this molecule in our body called interleukin-6 or IL-6. Basically, it stimulates these satellite cells to divide, which causes your muscle mass to increase. So the opposite of hypertrophy, the opposite is hypo. We actually don't say hypotrophy. We actually say atrophy. So atrophy would be a decrease in size and strength of skeletal muscle. Basically, if you don't use your skeletal muscles, you're going to lose them. Your actin and myosin filaments, they're going to decrease in size. The number of mitochondria that you have, your body's going to get rid of it because you're not using it. Aerobic exercise actually stimulates your slow twitch fibers, which makes sense because you're not doing a lot of fast running with aerobic exercise. Forceful exercise stimulates your fast twitch fibers because you're doing a lot of fast moving. So that's going to stimulate fast twitch fibers, which is going to produce more actin and myosin. All right, so moving on to smooth muscle. So when we talk about smooth muscle, we'll compare it to skeletal muscle. Smooth muscle is actually shorter in length than skeletal muscle. Remember, skeletal muscle can be up to 20 centimeters, which is almost 8 inches. Skeletal muscle has a lot of nuclei per myocyte. Smooth muscle only has one nucleus. It's very centrally located. Smooth muscle kind of tapers off at the end. Smooth muscle is under involuntary control. That's your digestive system, your internal organs. It contracts and relaxes more slowly than skeletal muscle. When we look at smooth muscle under the microscope, it doesn't have that striated pattern that skeletal muscle has. It lacks transverse tubules. That sarcoplasmic reticulum is not well developed. Remember that I said that the T tubules in the sarcoplasmic reticulum of skeletal muscle and cardiac muscle is well developed, but not so much in smooth muscle. When we look at smooth muscle, we can classify smooth muscle as either visceral smooth muscle or multi-unit smooth muscle. Visceral smooth muscle kind of acts as a unit, like your digestive system. These, remember, gap junctions allow for cell-to-cell communication. We'll see this in a lot of our hollow organs, your digestive system, small intestine, large intestine, stomach. So it's very common in smooth muscle. It says exhibit rhythmicity, peristalsis. That's what moves the food along your digestive system in that rhythmic pattern. And your cells have to communicate to say, hey, I contracted, now it's your time to contract, now it's your time to contract. And that's what moves your food along in your digestive system. Multi-unit smooth muscle. This is where your cells kind of function as kind of separate units as opposed to an organized system. They're really not organized into sheets, but they're going to function more independently. So the muscles of your eye that contract and dilate your pupil, they don't really interact with any other type of muscle. Your muscles of your blood vessel walls that vasoconstrict and vasodilate your arteries or your arterioles, they really don't interact with any other type of muscle. So they're going to function kind of by themselves in response to needed for blood pressure changes. They're going to be stimulated by neurons or by hormones. So they're going to be kind of standoffish and function by themselves. All right, smooth muscle. How smooth muscle contracts kind of resembles how skeletal muscle contracts. Both use actin and myosin, both use calcium, ATP. They're both triggered by that membrane, that neuromuscular junction, but there are some differences. Instead of, remember, skeletal muscle has that troponin, tropomyosin complex. Instead of troponin, smooth muscle uses this compound called calmodulin. When smooth muscle contracts, skeletal muscle, we have acetylcholine. For smooth muscle, we have two neurotransmitters. One's called acetylcholine and one's called norepinephrine. And we'll talk about this when we talk about the parasympathetic and sympathetic nervous system. The sympathetic nervous system, fight or flight, will be acetylcholine, and the sympathetic nervous system, fight or flight, will be norepinephrine, and the parasympathetic nervous system, or the rest or digest, will be acetylcholine. We'll talk about that in a subsequent chapter. And like I said, hormones can either stimulate or inhibit smooth muscle, depending on if it's parasympathetic or sympathetic nervous system. Stretching can trigger smooth muscle contraction. So think about when you eat. When you eat, your stomach gets bigger because of the food that you ingested on Thanksgiving Day, and that stretching of the stomach will actually cause certain hormones to release and signals to your brain, and that will say, hey, my stomach is full. Let's dump out some food into my small intestine. We'll talk about that next semester. Smooth muscle is more resistant to fatigue. You can only work out your skeletal muscles for so long before you're exhausted. Smooth muscle works not 24/7, but a lot longer than skeletal muscle. All right, cardiac muscle. Obviously, it's only found in the heart. Cardiac muscle, these muscle fibers are joined by these things called intercalated discs. And that's actually, we talked about the peristalsis in smooth muscle. These intercalated discs are for, and in cardiac muscle are for, so that the heart can beat, the atria, ventricles, so that they can work as a unit. I should put this one up here, so like that. So that those fibers can contract as what's called a syncytium. Oops, edit, undo. All right, they contain a single nucleus. So smooth muscle, cardiac muscle, single nucleus. Skeletal muscle, multiple nuclei, multinucleated. Cardiac muscle, self-exciting. We say it's automatic or automaticity and rhythmic. It doesn't require stimulation by motor neurons. So I could actually dissect you, take out your heart, and it will continue beating for a certain amount of time. We have these automatic pacemakers inside of the heart. They have their own kind of nervous system inside the heart. If you've ever heard of your SA node, your AV node, there's internal pacemakers when you're within your heart that will allow your heart to beat. But yes, there is nervous system stimulation, motor neurons that will stimulate your heart as well. Longer refractory period than skeletal muscle. We'll talk about refractory period next time or next chapter. Remember those transverse tubules and sarcoplasmic reticulum of skeletal muscle and cardiac muscle cells are well-developed. Smooth muscle, they are not. Here are the picture of the intercalated discs of the cardiac muscle. All right, just a summary slide of the three different types of muscle tissue. You should be familiar with those. All right, skeletal muscle actions. We all know that skeletal muscles generate different types of movement. Action of the muscle depends on what type of joint it's associated with. That the different type of joint, your ball and socket joint, some joints only allow for flexion and extension. Some joints allow for rotation, multi-axial movement. All right, body movements. So we're not going to spend too much time on levers. That's physics. So bones and muscles function together as these mechanical devices called levers. We have four different basic components of levers. Basically, we have these bars or rods, which are actually our bones. We have a fulcrum, which are our joints, and that's what those bones pivot on. We have a weight or the object that provides resistance, and then our muscles would be the force. And really, the only thing you need to know is that the elbow joint acts as a fulcrum. We have first-class levers, second-class levers, third-class levers. We're not getting into all of this. Just memorize the underlying things, and we'll go from there. All right, we've already talked about origins and insertions last chapter. The origin is the less movable end. The insertion is the more movable end. And when you contract the muscle, you're actually pulling the insertion up towards the origin. Last chapter, I talked about the biceps brachii. Here we're talking about the triceps brachii. Tri means three, so it has three origins. So remember, insertion is pulled towards the origin. All right, so when we contract a muscle, we're actually not, when we contract the biceps, that is called the agonist muscle. That's the muscle that is causing or that causes an action. However, we are not only contracting the biceps. We are actually using synergistic muscles, muscles that assist that agonist or prime mover. And I guess I should say, sometimes we'll call the agonist the prime mover as well. We also have what's called an antagonist, and each muscle has an antagonistic muscle. And that's just a muscle whose contraction is in the opposite direction of that prime mover or the opposite of the agonistic muscle. So typically when you're working out your bicep muscle, which is your agonist muscle, the triceps is going to be the antagonistic muscle. So if you're working out your bicep, you're going to have to work out your tricep as well. Muscle nomenclature, we're actually not going over this in class. We'll cover this under the lab portion. Myasthenia gravis. So that is an autoimmune disorder. Autoimmune, remember, the body attacks itself. Basically, the body is attacking those acetylcholine receptors on that motor end plate in that neuromuscular junction. So essentially, a person only has about 1/3 of the number of normal acetylcholine receptors. So they're going to have a decreased number of acetylcholine receptors. So because they have a decreased number of acetylcholine receptors, you're going to have muscle weakness, a lot of muscle fatigue. And what we can do is give them drugs that inhibit acetylcholinesterase, give them immunosuppressant drugs to decrease their immune system so that hopefully they don't have those antibodies to attack those acetylcholine receptors. You can give them antibodies that hopefully will hopefully attack their the antibodies that are attacking their acetylcholine receptors. You can also do a plasma exchange. But I want to talk about drugs that inhibit acetylcholinesterase. So basically we have acetylcholine and we have acetylcholine esterase. And acetylcholinesterase, remember, breaks down acetylcholine. So if we can have a drug or a medication, and I'm actually going to change this to medications that inhibit acetylcholinesterase. So if we have a medication that inhibits acetylcholinesterase, if we get rid of acetylcholinesterase, we will leave acetylcholine in that synaptic cleft longer in hopes that acetylcholine can attach to those acetylcholine receptors that are left in that synaptic cleft. So that's why we would give a medication that would inhibit acetylcholine esterase. All right, muscular dystrophy. Basically, we have a deficiency in a muscle protein called dystrophin. And that dystrophin binds to that sarcolemma, and basically the cells lose their normal structure, and they kind of die off. Clostridium botulinum, think of botulism. That's a bacteria, an anaerobic without oxygen. So it's a bacteria that thrives in a low or no oxygen environment, and it produces a toxin from food poisoning. Found in food that's not properly preserved. It can cause digestive muscular respiratory syndromes or symptoms. It is treated with an antitoxin, but we use Botox to actually smooth wrinkles, treat migraines. Remember, we had that question about hyperhidrosis and using Botox with that. All right, TMJ, temporomandibular syndrome. We already talked about that. And the dentist checks your teeth, so we don't need to go over that again. So I'm actually going to delete that slide here. All right, lifespan changes. As we get older, the amount of myoglobin, remember that's the hemoglobin in your muscle tissue, ATP, creatine phosphate, that all starts to decline. Your connective tissue and your adipose cells, those are your fat cells, that replaces some of your muscle tissue. Once you reach about 80, about half of your muscle tissue has atrophied. You've lost about half of your muscle tissue. Your muscle strength decreases. Your reflexes become slower. However, just as with your joints, exercise, activity is going to help maintain your muscle mass and your function. Oh, I love this question. All right, rigor mortis. I think you all know what rigor mortis is, or have at least heard of it. Rigor mortis, it's what happens after you die. It's basically your body becomes stiff a few hours after death due to chemical changes in the myofibrils. What do you think would cause the stiffness? There's an increase in ATP in the cells. There's an increase in ATP in the cells and a decreased membrane permeability to calcium. There's a continued neural activity that produces sustained contractions. There's a decrease in ATP and an increased membrane permeability to calcium. All right, so think about what happens during skeletal muscle contraction. So we have calcium. All right, let's just go through the answers. There's an increase in ATP in the cells. So what produces ATP? Mitochondria. How does mitochondria produce ATP? Glycolysis, citric acid cycle, electron transport chain. Well, if you're dead, you can't produce ATP. So here, you're dead. There's an increase in ATP in the cells. You're dead. You can't produce ATP. There is continued neural activity. You're dead. You don't have any neural activity. There's a decrease in ATP and an increase in membrane permeability to calcium. So you do have a decrease in ATP. You're dead. You don't have any ATP left. And what happens when you die is that your cell membrane starts to, one, disintegrate, and two, remember your cell membrane has all these channels and these gated channels and these ion channels that allow for that selective permeability. It can only allow certain ions to cross and certain things to cross over. Well, now that you're dead, it can't tell, it can't say, oh, well, I have a concentration gradient, or you can't come in due to polarity. You can't exit the cell. You can't do this. You can't do that. So that membrane permeability really is no longer there. So now a lot of your ions can just freely cross back and forth. So you actually have an increase in your membrane permeability, especially to calcium. And remember, calcium can now freely cross that cell membrane, and calcium can still bind to troponin, still move troponin out of the way, still move tropomyosin out of the way. And that's what causes rigor mortis. And I was actually a CNA working in the ICU, and we had 4 patients, and one of them had passed away. And it was going to be about 6 hours before the ambulance could get there to take her to the funeral home. And this lady sat straight up in bed, and it scared the living you-know-what out of me. And it was rigor mortis. So anyway, so D is going to be our answer. We don't have any ATP because we're dead, but we do have an increase in our membrane permeability because we don't have anything stopping calcium from entering into those myocytes, that sarcoplasmic reticulum, those transverse tubules, and calcium can still stimulate those muscle cells to contract. All right, so that's all I have for chapter 9. Please let me know what questions you have, and I will see you next chapter. Take care.